look at plants.
Hello, everyone. Thank you for joining the Science Talk show presented by Times of Biotech. Today, we have with us Dr. James White, Professor of Plant Biology at Rutgers University, New Jersey, USA. So with no further ado, let's welcome Professor James White. Hello, Professor. Welcome to the show.
Hi, Sobin. It's nice to meet you. Thank you very much. I'm happy to be here.
Sir, pleasure is mine to have you on the show. So, you are well known for your research on endophytic microbes of plants. But before we go ahead and talk about the research, first I would like to know your motivational journey to success and what inspired to come to this research field.
Well, what first got me started was as an undergraduate student at Auburn University in Alabama. I got involved with some research on fungal endophytes and in grasses, and that it was so exciting to be able to look at plants and discover they had these little hidden microbes inside them that really that turned me on, and I never lost that excitement of being able to find those microbes and study what they're doing. So for a good 40, almost 43 years now, I've been working on these endophytic microbes in plants.
Thank you for sharing your story, sir. Another thing is that I want to like to know, I'm really curious because I have read that you are well known for the endophytic microbes of plant research. So I'm really curious to know about your research. And specifically, I also got to know about the Rhizophagy cycle, which you developed in a collaboration with your students. So I would like to know more about that thing, sir.
Okay, I will share a slide set then and go over a few items that we're working on that are really exciting to us right now. So let me see if I can do that. And good. Okay, so endophyte journey, I called this. And I'm not used to talking about myself, so I'm not going to say too much about myself. But as I mentioned, I've been working on endophytes in plants for a good 43 years. And that really started as an undergraduate at Auburn University back in, I graduated in 1981, so it started actually 2 years earlier from that, the work did. And through the University of Texas, I worked on endophytes and got my PhD there. Postdoc at Ohio State, I was looking at fossil fungi. Assistant professor at Auburn Montgomery, still working on endophytes, through full professor here at Rutgers, working on endophytes. Bacteria, fungal endophytes at first, then bacterial endophytes later. And it's been a fantastic journey looking at endophytes and trying to figure out how they can be used in agriculture. So it's really an exciting area. And I should say, I now have worked 43 years on endophytes. Also have a family, and this are two of my wife and I. Shari is my wife, and our granddaughters are shown here. So River and Amaranth. So you see, there have a whole family too. And here's my wife with our two grandbabies. After the first one was born, this is Shari. So my wife of 42 years at least going on. So these are endophytes. This is in grasses. And you can see the blue here to the left. That's the fungus inside a grass with tall fescue. You see the grass over to the right. Endophyte meaning inside, endo meaning inside, and phyte meaning plant. So any microbe that goes into plants. And okay, so all these plants, all plants have endophytes in different parts, and really bacteria are the most common kinds of endophytes in plants. And you see whole communities of bacteria going into plants. Here's a root hair of a plant, and you can see the little blue dots in there. Those are actually the bacteria that get absorbed into these root hairs, into root cells from originally from the soil, but then they're seed transmitted once they adapt to the plant, so the plant can pass these on through the seed. Endophytes are everywhere. That's the good thing about working on endophytes. They're everywhere, and what they do is really largely unknown. I found this is really virgin territory in terms of trying to understand what's happening inside plants. It really is almost everything we do is brand new. It's interesting. Crops have endophytes. We think a lot of crops have lost them, so we're interested in studying wild plants so we can figure out what's happening and put them back in the crops. That's our thought. But some crops, they definitely have endophytes. Here's one. This is my graduate student working on hemp. April Michi working on hemp. And if you look at the hemp seeds, they're actually full of endophytes that when the seed germinates, they go all in there. This is a root hair from the hemp seedling. You can see the little dots here to the right, picture to the right. Those are the bacteria inside those root hairs. These are hemp root hairs. And, they're invisible unless you know how to look for them, unless you stain them with specific types of stains, and then you can visualize them. So most people don't. Many people have lost the ability to, they don't know much about microscopy use, and they're not able to, don't have microscopes that are good enough to visualize these endophytes in plants. But with a good microscope and the right stains, you can see these reds in every plant. Okay, this is one of my students, also Celeste Zong, and what she did is she labeled one of her Pseudomonas endophytes with a fluorescent probe called M. cherry. Then she looked at it with a confocal microscope, and the M. cherry is on the bacteria, and you can see now inside these cells all these little red dots that keep appearing as it scans through this image. And those are, the blue is the cell wall of the cell. You can see those are inside underneath the cell wall. See the red dots underneath the cell wall. They're actually inside the cells. These cells happen to be root cap cells that slough off, that fall off the tip of the root. But they're in all the cells of the root. This is just another one, and you can see the bacteria in there, just like these little moving things that Celeste has made. And you can confirm that, in fact, those bacteria are inside. And this kind of work was done back in 2010 when they discovered these bacteria inside root cells. And there's a group in Queensland, Australia. A young lady here to the right, Chani Pongfu, is one of the team members in Queensland, Australia. And they found bacteria inside roots of plants, tomato plants. They labeled those bacteria with GFP, and then they put them in tomato, and they put them in Arabidopsis. And you can see them in the root hair. The image here to the left, you see the little green dots. Okay, and then this Queensland, Australia group named this process rhizophagy cycle. Rhizo for root, phagy for eating, rhizophagy. And that denotes the idea that these plant roots are actually absorbing soil microbes and then degrading those soil microbes as an energy or a nutrient source. So rhizophagy. And that was 11 years ago. Time flies. We've been working on it since then. And what we found is that it actually is a cyclic process. And that is that microbes are attracted from the soil. They're attracted to the root tip because the roots are secreting exudates. So they follow to the exudates. And then at the root tip, they're internalized into root cells. They go into the cell, and the plant uses reactive oxygen or superoxide to extract nutrients from the microbes. And then the surviving microbes are actually trigger root hair development. They'll produce, the microbes produce ethylene, and then that will trigger root hairs to form. And then the surviving microbes, as those root hairs form, it'll squirt them, eject those bacteria out of the soil from pores in the tips of the hairs. And so they'll go back in the soil and acquire more nutrients and attract it back to and so forth. So you have this cycle. If you look over here to the left, you can see bacteria coming out of a hair tip. See it right in there. This is a fluorescent stained bacteria. So bacteria, they're emerging from the root tip, from the root hair tip back to the soil. They actually, for the most part in the rhizophagy cycle, they don't go into the cytoplasm. Instead, they're out in this periplasmic space, and that's just underneath the cell wall, but outside the membrane. But this is where the plant can put reactive oxygen, superoxide onto them, and extract nutrients from them. The plants are also moving the bacteria around in the cell, and we think that enables more nutrient extraction, more efficient extraction from the microbes in this process. And this actually shows a hair below. You can see the root hair, and the red, this is stained for hydrogen peroxide, another form of reactive oxygen, and it's around the bacteria. So the bacteria are out in this periplasmic space, and the plant is secreting this reactive oxygen onto the microbe. But at the same time, it's moving them. Now if you look above, now you see this cyclosis movement. So these shadows that are going around are these clusters of bacteria that the plant is moving. So we think of root hairs as just static things just sitting in the soil, but in fact, they're dynamic. They're moving, they're extracting nutrients from these microbes. And in fact, we think of root hairs as merely, passive little extensions of the epidermis that go into the soil and absorb nutrients that are dissolved in the water. That's actually a not accurate characterization of what root hairs do and how they work. Root hairs in plants work to process microbes and absorb nutrients from those microbes, but also absorb nutrients from the soil and the liquid part of the soil, solubilized in the soil liquid. But another thing that they do in root hairs is they actually will multiply these microbes and make a bunch of copies of them, clones. And then when those root hairs elongate, they squirt them back into the soil. So the plant is essentially repopulating the soil with these microbes that work with it and carry nutrients back to it. So plants are really processing microbes, symbiotic microbes, with these root hairs. It's a very important microbial processing function. This shows a root hair, and you can see the cyclosis. You can see the bacteria moving around, but you see they accumulate in the tip of the hair. You see the little white dots there. What happens is in this tip, all those microbes in mass will secrete ethylene, which ethylene is a growth hormone that plants use to grow, and then as because of that ethylene secretion in mass, this root hair will elongate just a little bit, how much actually it is probably 5, 6, 7 micrometers or something like that in elongation. But that's enough. As that elongation happens, that's enough to squirt these microbes out of the tip of the root hair, and then the hair starts over again and starts building up and replicating these microbes until it gets a cluster at the end, and that produces ethylene and causes another elongation and another growth spurt. It's really, really cool. This shows actually one of these root hairs. They get fat with these bacteria. You can see some of the red stains and some that are not, but actually all of this white area here is all full of bacteria. It's just not staining with this hydrogen peroxide stain that we use, this particular stain. But you can see where the bacteria came out of the tip. And they come out of the tip there, and then they reform their cell walls. And this work, this is my granddaughter. River, the granddaughter River, and she's out doing agriculture now. You can see her, you can see some compost there. This process in which plants are cultivating microbes and extracting nutrients from soil microbes is important in the field of regenerative agriculture. And that is, they have always, in regenerative agriculture, they've always thought that soil microbes are important. And so this is one way that soil microbes are important, that plants are actually absorbing those soil microbes to get nutrients. So, there's been a lot of interest in rhizophagy cycle over the past few years because of the in regenerative agriculture. Well, more recently, we are studying nitrogen fixation in plants and trying to figure out if plants are fixed in nitrogen. You can actually do these. We've done a lot of these nitrogen 15 tracking experiments, and you can get wild plants and then put them in a chamber of gas, of nitrogen 15 gas, and let them grow a little bit. Let them remain for 7 days or so for a period of time, and then analyze the tissues. And what you can see is that many of these wild plants are absorbing nitrogen 15 from the atmosphere. And so and we know that most plants can't do that. I mean, only legumes and certain ones that have rhizobia, root nodules do that, but many plants that don't have nodules, and many different families of plants are also doing that, absorbing nitrogen from the atmosphere. And so we've been trying to figure out, how they're doing it. And what we've come to appreciate now, after studying for more than a year now on plants, is that are fixing nitrogen like this, is that there actually appear to be the trichomes on plants and other non-photosynthetic parts of plants contain bacteria. Trichomes are plant hairs, and they're all over leaves, typically on the lower surfaces of the leaves. And they will contain bacteria in their structures that we have evidence are fixing nitrogen and transferring that to the plant, and just a little bit of that. And this is some variation in trichomes. And plants have all kinds, and people used to think, well, trichomes are defensive. The problem with the defensive theory of trichomes is that insects just eat right through the trichome. They're just the soft structures that make them bite through the trichome. Some of them filled with chemicals, and people have thought that those are defensive. In some cases, some animals don't consume them because of the chemicals in them, but in other cases, animals just eat them right away. There's no benefit. There appears no defensive benefit. The defensive theory doesn't hold up either very well. But okay, here's one example. This is hemp, cannabis, and this is my student in April Meach, who's been working on this. And these trichomes are on the leaves, but they're also on the fruits, or the bracts on the fruits. And so this is an illustration of these trichomes. And this is a glandular trichome of cannabis. And it's composed of plant cells. And it also, what we're finding is it's also composed of bacteria. Okay, this is an actual picture of it. I'll note the structure. Note it has a head, and there's no chlorophyll in that head. There's no evidence of photosynthesis there. So it doesn't have photosynthesis. And you see the base, and the base, you can see this big base, it's kind of green. It has photosynthesis in that base. So, anyway, it's so cool. These things are so cool. Okay, this is one of these trichomes looking down on it, and this is stained for superoxide, which is a purple color. And the superoxide forms around bacteria and in there. And so you can see the purple, and the red arrows are showing the purple. That's where bacteria are in that. This is a young trichome just developing, and this trichome these microbes will replicate in there. We think that the trichome itself is replicating these microbes. Here's a closer up look of that, and you can see the rods actually inside, little clusters of bacteria inside those developing glandular trichomes of cannabis. a lot of plants have glandular trichomes, and they all have bacteria in them. We have looked at some, I don't know, 13 different plant families at this point, and most of those plants have trichomes that are either filamentous or they're glandular, and for the most part, all but 2 of them appear to have bacteria in those trichomes. So it's this actually is a hemp glandular trichome again, and here you can see the plant cells in the middle, see the little plant cells, and you can see all of this around the periphery. We think all of this peripheral is actually bacteria. And you can see interfacing the bacteria and the plant. You can see the blue. That's superoxide. Oops, that's superoxide in this stain. And what's happening is the plant cells are secreting superoxide onto those bacteria to get nutrients out of them, like in the root here that I talked about. Yeah, so they're full of bacteria. And what we think is happening is this. If you look at the side view again, you get photosynthesis in the base, and that's where sugars are produced, and it transports those sugars up into the head. And that feeds the microbes in that head sugars that they need in order to fix nitrogen using nitrogenase. And in fact, we think that what the chemicals, the terpenoids, like THC that's in hemp trichomes, glandular trichomes, and other glandular trichomes are actually antioxidants that reduce the oxygen from the atmosphere, because atmospheric oxygen will actually suppress nitrogenase activity. So we think the plant is producing those chemicals not to make us high or to screw up, the thinking of insects that may consume this plant, but rather to actually scavenge oxygen and make it so that nitrogen fixation can happen more efficiently. So that's we're working on that. The big paper actually that's involved a lot of study, and that'll be part of that big paper right now. Probably come out the first of the year or so. Expect to submit it at least the first of the year. This is another plant, and it'll show another kind of trichome. This is tree of heaven plant. And it's invasive, grows like a weed. We have it all over New Jersey now and all over the United States. I think it came initially from Asia. You might have them in India, I'm not sure, but they're certainly all over the United States.
I have seen such, sir, but I'm not very much sure about it.
It's a beautiful tree. It's a beautiful tree.
Right.
They're aggressive. They grow in cities. They're like weeds. It's hard to kill them. Well, if you look at the leaves, you can see these are the kind of trichomes they have, the filamentous trichomes. Beautiful big filaments. And if you look closer at those trichomes, this shows them, you can see the little dots in there. And you can see coming out, little pores there. You can see the little chains of bacteria coming out of those trichomes. These are loaded with bacteria. And they are, in this case, we've isolated a bacillus from some of the leaf washes here. It's hard to kind of say, okay, well, this dot in here is actually the bacterium, this bacterium that you get out, because we're not very good at micromanipulation and reaching in and grabbing a bacterium from inside a cell. It's really hard. This is hard, difficult work to do. So you kind of, but anyways, here you see bacteria coming out, and you can actually see that white here, the upper arrow, that very white, that's an endospore. And you can see to the other lower arrow, you can see the very white ones. Those are endospores. This is from this bacillus that is coming out of these hairs. Anyway, it's really interesting. There also is another, not only are trichomes active, but we have in some plants like desert plants, the hosta. Hosta is one of the desert plants that doesn't have trichomes. We think because the trichomes would be thin-walled and they would leak too much water for these desert plants. So they've dispensed with trichomes. But instead, I mean, if you are familiar with hosta, they have yellow areas, green areas with photosynthesis on their leaves, and they have areas with no chlorophyll in them. And if you look at the areas with no chlorophyll and you look at the epidermal cells in those areas, you'll see that they have bacteria in those cells. And this is actually one of those. You can hear right beside them. You see to the bottom part, you see chloroplasts in the cell, and then you see this other area with no chloroplasts. And in this other area with no chloroplasts, you can see the nucleus of the cell there. You see the little dots in the nucleus. This is stained for reducing sugars using copper sulfate. And the kind of orange coloration you see in there is due to sugars like glucose or fructose being present inside there. And we think that the plant is putting some sugar in these nuclei. I know it sounds very strange, but we've seen it in several plants now. And some plants may be converting in some cell types. They may be converting their nuclei to areas where they can cultivate bacteria, nitrogen fixing bacteria. This shows a nucleus. Now this is stained for using nitro blue tetrazolium, stained for superoxide, which makes it blue. You can see a nucleus, and you can see the bacteria come out. You see all around, and you see those are actually vesicles that come out of those nuclei. It sounds so crazy. You guys are going to think I'm nuts. But in time, you're going to see that in plants, this is what plants are doing, so what's happening here is the plant is reproducing these microbes, giving them sugars, enabling them to fix nitrogen inside the nuclei. Then at some point, it releases them from the nuclei into vesicles that go into the cytoplasm. And once it goes into the cytoplasm, then you see the superoxide start happening. So when it's in the nucleus, you don't get superoxide. When it's out, you get superoxide. So
Yes,
can I show you some more? Yeah, isn't that crazy, Sophie?
Sorry?
Isn't that nuts? It's very strange, isn't it?
Yes, sir.
Okay, I'll show you again. I'll show you some more. Okay, here now is nitrate stain, and you can see the nucleus here, and you can see some bacteria in there, and then you can see they're coming out in vesicles now, and you have little clusters coming out of the vesicles, and this is stained for nitrate, which stains blue where the nitrate is. And you can see the nitrate is more concentrated in these vesicles that come out of those nuclei. You can see it over here to the other one. And here, actually, this is showing the same thing. This is nitrate again. You can see in the nucleus, you can see there's not much nitrate. You can see the bacteria dots in there, but you can see when they come out now in these vesicles, you can see it's blue. And these bacteria come out into the cytoplasm. And I'll show you, we actually have a film. Okay, this shows more movement out of these trichomes, bacteria out, and you see when they go out there, they begin to be blue. And what's happening actually is once they leave the nucleus, the plant detects them and then shoots superoxide on them, triggering them to then secrete forms of nitrogen, antioxidant forms of nitrogen to protect themselves. And then that gets, like nitric oxide, for example, and then that gets transformed into nitrate once it's oxidized by the superoxide. So, and we actually have some movies of this. This is a movie. This is a nucleus, and it is a hosta. And you can see if you notice up here at the top, see bacteria coming out little by little.
Moving.
They're moving, but they're actually more of them are appearing in this area. You see they're getting more and more, and you can also see a little bit. You can see there, at least in some of them, you can see. Now, I see another one just came out. Did you see that? Right in there, it just came out. So it just came out. So they're emerging from the nuclei, and when they emerge, the plant hits them with superoxide, and they start to they start to produce nitrate, and the plant then extracts them for nitrogen once they go into the cytoplasm. So it's a cool system. I mean, this is a system for extraction from microbes, really, that's based on the innate defensive system of eukaryotes that eukaryotes use as defense. They use reactive oxygen, they use superoxide. So this same system that happens in our cells and happens in plants for defense is converted into plants to use for extraction of nutrients from microbes. Yeah, so here's another one that just shows these bacteria coming out, and you'll see little ones, nucleus in the background, and you'll see them. I'm not going to show this whole thing, it goes for a while, but they're coming out here and you'll see them appearing.
Yes. And it's quite clear. So.
It's pretty cool.
Yeah.
It's a cool thing. That shows little ones coming out. You can see they're tiny little vesicles here, but they're emerging from that nucleus. Those are the bacteria. And of course, there's DNA in there. We don't know anything about whether the plant's DNA is intact or whether these will become, we just don't know what's happening inside these nuclei. We can see the microbes coming out. We can see them being oxidized, but we don't know any of the details there. So anyways, here's some references. You've seen the previous work that we've done, some of the previous work on Rhizophagy cycle, and you see our current work that we've done. And here's some articles. These are open access articles if anyone's interested in looking up some of this. The histochemical tests that we use are coming out of this paper that we wrote. We had to invent a lot of these histochemical tests ourselves in the lab in order to examine what's happening microscopically between the microbes and the plants. And this comes out of this paper, Chang, Kingsley, and White. This was actually a 2021, just this year, earlier this year that we did that. So, and so that's hot off the press. And you see pre-press stuff. We're still developing the paper, and we're not too worried about being scooped by it, because it's it's really difficult to do this work, and most people, you have to have a really good microscope, and you gotta, it takes a lot of data to put together to get the story of what's happening. So I mean, actually, we want more people working in this area, because we think that this is a very significant phenomenon happening in all plants, and that it could be very important for agriculture, and that especially for future agriculture, where we're going to grow crops, for example, with no nitrogen fertilizer or minimal nitrogen fertilizer, right? Then taking advantage of these nitrogen fixing endophytes and being able to tweak that system, to make them so they fix more nitrogen in these trichomes and other cells, not photosynthetic cells, that would be, that would save the environment basically from all the nitrogen runoff that we have. So we think this is a very important process. And so anybody who wants to work on it, we invite to do so. We will help them in any way that we can. The groups are doing that, but it takes more really to understand this kind of phenomenon. So these are all the people in the lab who did this work, who contributed to this work, and there's a lot of groups, postdocs and students, and so I'm just going to stop.
Yes, sir. I think that was a very resourceful presentation, and I would like to tell all my viewers that if the people thought that this was really a beautiful phenomenon, because I love it, and the way sir have explained it is like I think it's very resourceful and the people can make use of it. And as sir said that very few people are working in this area. Of course, I was also not aware much about this, but after sir's presentation, I think this is a beautiful area of nature's beauty to be explored, and it can be used because we have a lot of problems related to agriculture and food security. And as sir said, that it can also be used for the current situation and solving a lot of problems and saving the environment. So I think this is a pretty cool thing.
I think it is pretty cool, and I agree with you. There is beauty in it, and these plants are doing things that we had no idea they were doing. The image that most people have for most plants is that they grow in the soil, they wait around for some kind of fertilizer to be put on it to grow.
Right.
But of course, that's so far from the truth, so far from correct. And the truth is, they are cultivating microbes, they're internalizing those microbes, and they're processing those microbes for nutrients. And it's systems that we never thought they had. They go inside the microbes. They take the microbes inside the plant cells, inside the cells of the plant. Imagine that, in the cells themselves. We never thought that. And this is just the concept of that is what blows a lot of people away. They get stuck up on the idea, oh, they're going in the cells. That's impossible. It's impossible. Right?
Yeah.
It's impossible.
Right.
Yeah. And
It could
beautiful.
be, it can be a fantasy to some people. So I think this has to be explored. So a lot of people can actually take inspiration from you on this, sir. So, I would like to tell my viewers that Sir is not only an amazing researcher, besides that, he has also played a key role in establishment of International Symbiosis Society, and also served as a founding secretary from 1992 to 1995. So can you please share some important points related to this foundation?
Well, yes, it was a long time ago now, but early in my career, there was interest in developing this was early in internet development, when the computer internet, was developed. And we had what's called news groups in those days. And news groups, there was one of them and they're just like a listserv where a few people were involved, right? And one of them was on symbiosis. And so I was one of the people in the listserv. And so in that news group, and we decided we wanted to form a society. So that's what we did. We formed the International Symbiosis Society, and there is a journal called Symbiosis that was actually already established by Margalit Galoun in Israel. And we adopted the journal Symbiosis for the International Symbiosis Society. And now this is Symbiotes, International Symbiosis Society, ISS, right, is ongoing, has been going on, and it has meetings about every 3 years, something like that. There's an international meeting people go to for it. And it's one of the things that I did, I form a society. But it's one of the things that I did. So people do a lot of things through life, right?
Okay.
Yeah. Yes.
I think viewers can go ahead and check out that society, because the website is also, we can give some link of the website in the comment. I think that will be better. So
yes, that'll be good.
Yeah. Okay, sir. Now I would like to hit another part of your versatile career. That is like, you are experienced associate editors and also a member of editorial board for several journals. So can you explain the editor's point of view about a strong manuscript?
Yes. Okay, so first point about it, and you're right, I do a lot of, I'm on a lot of journals. I've edited a lot of papers in my life. I've written a lot of papers in my life. It's always work. Nothing is easy. If you make a little short article, it's easier than making a long article. But of course, the longer articles are more valuable, right? They're more into it, a lot more work. It's harder to do, but also some research requires a longer article. So the first thing that anyone notices about an article is that it is well written. Okay, and so and what I mean by that is, I mean the English part has to be really well written. I mean, it doesn't have to be perfect English, but it needs to be proof edited by someone who's really good, And sometimes if you have to pay someone to proof edit, it's very expensive. Now, I don't know what proof editing costs, but journals nowadays will have these proof editors. So, I mean, the language editors, right? So you could do that. Titles are very important. The title especially and the abstract especially have to be impeccably written. I mean, like no awkward wording in the title, right? No awkward wording in the abstract, because the editor, or associate editor, the first thing they'll read is the title and they'll read the abstract. And they're not going to read most of the rest of the paper. They'll scan the rest of the paper, maybe spot read here and there before they send it out. But if there's a problem with the title, or there's a problem with the abstract in conveying ideas and language, right, then they just reject it out hand or send it back and say it's not ready or something like that. Or if reviewers do get it, they'll say it's not well written or something. It affects, there's a bias there, right? There's a bias there. And it's not fair because not everyone is a native English speaker. Okay, but nevertheless, that's what happens. That's how it works. Now, the other thing that you have to do is you have to make sure that the article is well organized. Okay, so you want an introduction that's not too long, but that is clearly kind of an introduction part. And then you want to make sure that the other parts, the results, and even the discussion, you sectionate, make sections, at least when you're writing it. You make the sections when you're writing it, and then you cover every idea. You cover the idea under a section, or organized section, right? And then you keep refining that section. You refine that section. So basically, you work it, you organize it, you work it. If in the process of writing it, you find there's pieces missing, right? So you say, oh, this is a weakness. I need to go and get an experiment, do that experiment and fit this in there. Then you do that, so you write it, you go and you, even if you're not done writing the paper, you realize now, you made a heading, something that should be there, you think, and you don't have enough data on it. So you go back and do a little bit more. So when you do enough, and you're testing yourself, whereas a reviewer would test it, but you test it first, and say, well, what's missing? If possible, give it to somebody, give it to someone else, give it to a colleague, someone who cares, right? Someone who will read it. Some people don't, just someone who likes you, and they may not know the area, or they may not put time into it. You want someone to really read it, or really take a look at it and give you their opinion about it, right? It doesn't make it perfect, but that opinion is gold, sometimes. Okay. Sometimes it can really help. Yeah, so, and other than that, and then the other thing is about publishing. You should not be afraid of rejections. Don't be afraid of rejection. Take a rejection as meaning that you can now have an opportunity to improve the article again some more. You get it back to work on it again. Hopefully for the journal, they've not rejected forever and said you don't ever want to see this again. You can go back to that journal, or you could fix it more, improve it more, and send it to another journal, right? You can do that, but every article requires work. It's a process to publish it. And it's not only you writing it, it's making corrections from other people, trying to make it better every time. And the joy is in the doing. right? The joy of it. I mean, yeah, you get a little brief joy when you get it accepted, you know, yay, hooray, we did it.
Yeah.
But it's over. It's over then. You got no more fun to develop it. You go on to something new, right?
Yeah.
So the joy is in the doing of the science and the expressing the ideas and so forth. And so if you have the right attitude, you will do better. Because there's never, I get papers rejected too. Not everything that I put goes straight in. You know, you have to just keep developing and developing, and never give up, because every time it's just a chance. You know, if you have an idea, you know, it's likely worthwhile. You know, and you know, it's worth your effort and the challenge and the struggle, and you know. So, anyways, that's my thought on it. You got to enjoy the process. That's the most important thing right here.
The key is to enjoy, right, sir?
The key is to enjoy.
Yeah.
Yeah, and any rejection or any revisions that come back, you have to enjoy the opportunity to make your paper better. You know, and you're always thinking about how I could have done that, how I could have made it better. And when someone comes back and says that, oh boy, you can not only make their changes to make it better, but what you thought you could make it better too, then you have a chance to fix it. But you make it even better. So yeah, and you have to enjoy it.
Thank you, sir. But sir, I would like to ask you one more thing. This is like kind of silly, so I would go on the silly mistakes. Have you noticed anything which repeatedly comes to you and that seems really silly mistakes, and the people who wants to get their paper published should take care of it? Is there any points like that?
Yes, there are. And it all has to do with language. And sometimes we, you know, any of us who write think that the way we write something is the best way to write it, right? And so, I mean, I've seen it where adjustments get made, or I'll suggest an adjustment to wording or something like that. And then the author, the main author of the paper, will think that, well, their wording is better, or they prefer their wording. But the problem with that wording is it was awkward wording. And so, because, you know, we all think our writing is great. Yes, we all think our writing is great. We all think our writing is great. But the fact is, we all can use improvement, you know, in whatever it is, the title, the wording of a sentence, whatever it is. And so it really is important not to be married to what you have written in that way. You know, you can convey the idea better, clearer, more concise. So, you know, you have to be willing to give up your particular wording and listen to whoever it is or whatever it is, co-authors or reviewers or whatever. And ignoring reviewers or editors is, you know, we do that at our own peril, right? In fact, one of my best articles was written with an editor. I mean, he was the editor for the journal, I forget the pest management. It was a review, actually. It was a review article, an endophyte review article published just a few years ago. And the editor was an Israeli guy. He was like 80, he's 80 years old or something, really cantankerous, really. I mean, he would be very critical of the paper, very critical of the wording. And I mean, I was making corrections and he was writing back, you ignored my advice on that, you know, on the thing. He was really, really very aggressive editor. Well, you know, in the end, he had his way and that paper got published. And And to this day, I still hear comments that this is one of the best review articles on endophytes that people have seen coming out in recent years. So I mean, it can make a big difference, right? It can make a big difference. Yeah, these editors can really be helpful to you, or anyone that you have looking at can really help. If you have something confusing in there, you should pay attention to that and see if you can fix it like that. Just don't ignore the good advice. And that to me is a silly thing that I do and other people do, and it doesn't benefit the paper.
So basically, editors are there to get best out of yourself.
I think so. That is what I think. And honestly, they're not out to get people. Journals need articles, and so they want articles. And so if the article has some merit to it that they see, and if they can possibly get some, major revisions on it rather than rejections, then they'll try to help you get it better, so they can be published in that journal. That's their job. I mean, they're not just kind of like filtering out, bad papers. That's not really their job. Their job is to get the best out of an article. And sometimes if they issue a rejection, it's out of their hands. For some journals, if you get one rejection, boom, that's it. They have to reject. It's almost automatic. So anyways, they really try to be helpful in my experience.
Yeah, I think that everyone should take that editors are, whatever comments they are giving, they are helping you to beautify your article, or get more out of that article. So everyone should take that. So I won't bother you much, but I think I have last two questions, and one question which I repeatedly ask to every guest, that is about what you see into a student when they come to you for a PhD or any other courses for masters or anything. So what exactly you look and what you try to extract from their potential?
And that's a really good question. Dependability is something, this is a kind of a basic human trait, right? Somebody you can depend on them to go in and do their best to do some work. The ability to accept some guidance is important. Occasionally there are students who can't take guidance, and they always want to do everything their own way, and they don't know how to do it, or there's issues there that make it so it's hard for them. So taking some guidance is important. The other things that are really important is being excited about the work that they're doing. That's really important, to get into their work, to be excited about it. You can't teach someone that they become excited and latch onto a problem and start pursuing it and exploring it, and loving to try to see how that fits in nature and how it can fit into agriculture, and looking at the pieces and try to see the connections and so forth. That's native in some people. And you want that. That's to be encouraged. And those are good graduate students who do that. Dependability in terms of work ethic is, you gotta put the work in for research. And it's like any other job. You got to be there, show up. You got to show up. If you don't show up, you're not going to make any progress. It's like, I would say it's nice to have brilliant people, but the truth is, most graduate students are brilliant, right? They're brilliant. The truth is that the hardest thing about science is being disciplined and putting the work in, even when you don't see immediate returns. This is the hardest part. Keeping yourself motivated, imagining things, finding the exciting things about what you're doing, being flexible enough to follow the problem that seems the area that seems to be working out, so you go that way. You know, anyways, it's you know, do they work hard? Are they interested in what they're doing? You know, enough to keep doing, and that's important. It's nice when you're able to get along with students too, and they, you know, it clicks. Sometimes it clicks with students, sometimes it doesn't click as much. Either way is fine, provided they can keep moving forward in some way. Right? None of us are perfect, and sometimes it's better. Sometimes the student has a better idea than I have, and sometimes it takes me time to come around and figure that out. You know, that it was better that they were more correct with what they wanted to do. And then you know, if you click, then you're able to work as a team and move forward. And that's a really good thing. So yeah, so that.
Thank you, sir. Thank you for your opinion. So last thing is, last but not the least, I would like to ask you, what message would you like to give to the world?
Oh, that's a great question. Okay, I love that question. Okay. So to the world, I would say things aren't the way we thought they were. Mm. They're not the way we all thought they were with plants and with agriculture. And there's hope for, you know, our humanity developing cleaner agriculture. Hope, you know, with the students working so hard, and you have in India, you have a lot of people looking at, you know, plant microbiome, how that can be used in agriculture. And with all that energy, there's going to be some really neat new microbiome type agricultural innovations that I think is going to change how we do agriculture, how we look at plants, how we look at soil, how we care for soils, how we care for plants. You know, it's a cleaner, interesting, very biologically oriented future in agriculture that is coming. So we, you know, look for that to happen. And if you're doing research, a graduate student doing research, or professor doing research, let's work together and build that new future, that better future for, you know, science and humanity. So that's my message.
Thank you, sir. Thank you for the beautiful message. And I thank you so much for accepting my invitation and giving me time for this show. And I must say, the people who will watch this show will get benefited from your extraordinary research, which and a really truly a different research, and you are trying to explore the nature's beauty, and which nobody, out of the imagination, actually out of the box, we have come up with that nitrogen fixation, mutual thing, mutualism of the plants and the microbes, and another thing about the defense system, how plants are using that defense system for the nitrogen fixation, and everything was really superb. And apart from that, you gave a really good message about the agriculture and all about the soil and even the editorship. I think very few people know from this point of view, because when the paper is rejected, everybody says that what I did, I had written a beautiful paper, it was very resourceful, but why did it get rejected? So I think now people will have some idea, and they will accept the comments in a more of positive way, instead of thinking that what has happened to my paper, what's wrong, instead of thinking that way. So I think this was a really resourceful interview, and thank you so much, sir, for your time. Thank you so much.
You're so welcome, Sobhan. It's my pleasure, Sobhan. You have a good night.
Good night, sir. Thank you.
Bye-bye.
Bye.